Good evening and a warm welcome to you all. I'm Alison Button-Sloan, the Advocacy and Community Engagement Coordinator with Melanoma Patients Australia. First, I would like to acknowledge the traditional owners of the land upon which we meet tonight and their elders past and present and those emerging. It's wonderful to be here tonight and to be holding the third webinar of our four-part Melanoma Symposium series. We have 300 people attending from across Australia, which is just amazing, and I thank every one of you for coming along. Now, first up tonight, we'll hear from MPA's CEO and then on to our two guest expert speakers and our patient speaker. To some housekeeping. Now, at the bottom of your screens, you'll find the Q&A function, which will give you the opportunity to ask questions anonymously. We'll do our best to answer as many of them as we can in the time space that we have. There will be two Q&A sessions, one after Professor Long and one after Professor McArthur. Also, to let you know, this webinar is being recorded and it will be up on our website in a couple of weeks. So now I'd like to introduce Victoria Beadle, the CEO of Melanoma Patients Australia, who will give you an overview of our services. Thank you, Victoria.
Thanks, Alison. Thank you all so much for making the time to join us for this very special evening event. This is the third of our 2020 webinar Symposium series, which is being designed by patients and carers on the topics that matter most. This webinar series is bringing the latest information safely into the homes of melanoma patients and carers right across Australia. I also note that amongst the close to 300 registrants that we have for the event, we have people joining us from across the globe. And this is a true testament to the calibre of our speakers this evening and the unique environment that we find ourselves in due to the COVID-19 pandemic. To everyone joining us this evening, a very warm welcome. Our topics this evening include immunotherapy, latest treatment updates, and clinical trials. I am delighted that we have our patient speaker, Narelle, here with us this evening to discuss her personal experience of being on a clinical trial, such a critical issue. These topics will all be covered with a COVID-19 lens. And despite us dearly wishing that COVID-19 would go away, it is likely we will need to continue to adapt to it over the coming months ahead and the challenges it poses to those affected by melanoma, and especially those accessing treatments such as immunotherapy or considering a clinical trial. I encourage you to have your questions answered here tonight. Melanoma Patients Australia is dedicated to ensuring that our patients and carers have access to the best and latest information to help support and navigate them through what is often a life-changing diagnosis of melanoma. We're a small charity that packs a big punch with no government funding, and we work really hard to deliver high quality support, advocacy, and prevention activities. We have a small staff team and an amazing and dedicated network of volunteers across the country who work tirelessly to ensure that no one affected by melanoma walks alone. I'd like to thank our speakers tonight, Professor Long, Professor McArthur, and Narelle, for their time and expertise, and to thank our sponsors, BMS and Melanoma Institute Australia, who've made tonight possible. I'd like to remind everyone here tonight that you do not need to face a diagnosis of melanoma alone, and there is help and support available through Melanoma Patients Australia, including the National Melanoma Support Line and a range of support groups. All of our face-to-face groups have gone virtual, making them a safe and accessible space. Our closed Facebook support group has over 1,800 active and engaged patients and carers providing peer-to-peer support to one another in what is a very unique environment. We've recently launched a brand new closed Facebook support group specifically designed for carers. We also have a telephone support group designed for people with metastatic melanoma. And just a little reminder that we have newly established a telehealth nurse service which has been generously funded by the Melanoma Institute Australia and supports patients in navigating the health system, accessing available services, and making decisions regarding their health. All of our services are free and can be accessed wherever you live in Australia. Please reach out, find out more, and be connected into our very special melanoma support network. Thank you all for joining us this evening, and I hope you enjoy our keynote presentations. Thanks, Alison.
Thanks, Victoria. So now to our first guest speaker. Professor Georgina Long is a world-leading medical oncologist who, through her outstanding achievements in translational and clinical cancer research, has transformed the care of melanoma patients internationally and saved thousands of lives. She is the co-medical director of Melanoma Institute Australia, MIA, and chair of Melanoma Medical Oncology and Translational Research at MIA and the Royal North Shore Hospital, the University of Sydney. Prof Long leads an extensive clinical trials team and laboratory at MIA with a focus on targeted therapies and immuno-oncology in melanoma. She is principal investigator on phase 1, phase 2, and phase 3 clinical trials in adjuvant and metastatic melanoma. And she is the author of over 300 peer-reviewed publications in clinical and translational research in melanoma. In recognition of her work, she was awarded an AO in the Queen's Birthday Honours List in June this year. And I would like to add that Georgina sits on MPA's medical advisory panel. A warm welcome to you, Georgina. I know many people are looking forward to your presentation. Thank you.
Thank you, Victoria. It's a real pleasure to be talking about something that I'm very passionate about, and that is melanoma and treatments for melanoma. I'll just share my screen. Here we go. To start off, I wanted to show you some photographs of some of the people we've treated at Melanoma Institute Australia. These are 3 men who've all been treated with immunotherapy. On the left, you have Adam and Josh and their 2 beautiful babies. And they had stage 4 melanoma and had wonderful responses and were actually able to go on and to have families. And on the right is another one of our patients who had 4 doses of immunotherapy, had a complete response and stopped therapy, still remains in complete response, and is enjoying a very high quality of life despite his 102 years. I'm sure he's one of the oldest patients ever treated in the world. But the point is that all 3 of them have done incredibly well with treatment and enjoy very high quality of life. And here in the left corner is Josh when the baby Mia, it took me 3 to 4 months to work out Mia was named after Melanoma Institute Australia, but what a perfect name. And she's a bit older there with the photo next to her. And that this would not have been achieved 10 years ago for Josh and his wife. So it's wonderful. However, not everybody responds. This is another one of our patients who, this is when he was first diagnosed with stage 4 melanoma with a little baby in his family. And as time goes on, the disease just rapidly progressed and did not respond to any treatments. And sadly, David died. So this is to remind us that we've got a lot more work to do, and we will be continuing to do that work until we reach 0 deaths from melanoma. And this is a photo of his wonderful family, and it brings me to tears to think that he won't see his girls grow up. So what I'll be talking about is drug therapy in melanoma, focusing in on immunotherapy and neoadjuvant therapy, which I'll explain what exactly that is. Before we begin, it's really important to understand the stages of melanoma. So stage 1 and 2 melanoma is when, and this is all about skin, cutaneous melanoma. Of course, there are other types of melanoma, and we're working very hard in those areas as well, and that's uveal and mucosal melanoma. But for the purposes of today and the immunotherapy, I'm going to be mainly talking about cutaneous melanoma. So stage 1 and 2 is where you have the primary melanoma, and that is Arnie Schwarzenegger there with his melanoma on his cheek. He did not have any lymph nodes involved. He just had the primary melanoma. I don't know how thick his melanoma was. That's determined in the microscope, but he was either stage 1 or 2 melanoma. Stage 3 melanoma is where the melanoma spreads to the local lymph nodes. So here you can see if Arnold Schwarzenegger had metastases to the lymph node, it would look something like this. And this is a patient with lymph node involvement. This is what we call large lymph node involvement. That's macroscopic, but it can be microscopic, only determined by sentinel node biopsy. And that is stage 3, where the primary melanoma spreads to the nearby lymph nodes. And then stage 4 is where the melanoma spreads around the body to various organs distant from when you had the first primary melanoma. So the lung, the liver, the brain, the bone, for example, and melanoma can go anywhere. The stages are subdivided, and as you go down in numbers, the risk of a poor outcome increases. About 85 to 90% of melanomas in Australia are stage 1 and 2 and are cured with surgery and do not pose any long-term problem for patients. About 10 to 15% of melanomas are stage 3 or stage 4. And at the moment, we have drug therapy where we use it in both stage 3 and stage 4. And that's largely or completely due to the clinical trials and the work we've been doing over the last at least decade now. And we do have clinical trials for stage 2 melanoma for drug therapy. So what are the drugs that we have and that we use in melanoma? There are 2 major types. We tend not to use chemotherapy. If you've had family members or friends who've had chemotherapy, we tend not to use chemotherapy. The major types of drugs we use are targeted therapy. These drugs directly kill the cancer cell and are tablets. And at the moment, the ones on the PBS are aimed at the BRAF mutation, which is approximately 40% of melanomas. So that's targeted therapy. They directly kill the cancer cells. The second major type of treatments are immunotherapies. And at the moment, our immunotherapies are focused on the T cell in our immune system. We all have T cells. They're very important. We have a lot of other immune cells, but our successful drugs so far are aimed at the T cell. And these drugs do not kill the cancer cell directly. They get your own immune T cells to kill the cancer cells. So that's a major difference between these 2 drug types. I'm now going to show you some survival data in advanced melanoma. That's stage 4 melanoma. This is not for all stages of melanoma. So for stage 4 melanoma, where the melanoma is spread around the body, these are what we call survival curves. So at the start of time 0, 100% of patients are alive and well. And we start a drug therapy at time 0, and we measure how well patients do in terms of being alive with time. And what you can see is those 2 classes of drugs, the BRAF/MEK or the targeted drugs versus the immunotherapies. The targeted drugs do very well in the first year. The most people stay alive very well in that first year. And the immunotherapies here are not quite as effective in that first year. And then this is what we call Yervoy or ipilimumab in the purple, which we don't use first line anymore. At about 1 year, the survival rate is around 70 to 75% of the original 100%, and very similar across these new drug classes. About 10 years ago, the survival was less than 25% for 1 year for advanced melanoma, so huge improvement. However, the pattern of the targeted therapies being the most effective changes, and we see that by 2, 3, 4, 5 years, a pattern emerges where combination immunotherapy seems to have the highest survival rate, followed by single agent PD-1 immunotherapy, followed by targeted therapies. So it switches around. Now, this is incredible improvements from 10 years ago, but still approximately 50% of people with advanced stage melanoma still die of the disease. So we have a lot more work to do and to achieve 0 deaths. And that is why clinical trials are so important. So now what I'm going to do is I'm going to talk about the immunotherapies and how they work. Basically, I think of the immune system as a tug of war. Your immune system can either be tolerant. Now, we don't want it to be too tolerant. We want it to tolerate itself, your own body, but we don't want the immune system to tolerate nasties like bugs and viruses, because that way we get uncontrolled infection. So it's a balance. We want it to be activated enough against nasties, but we don't want it to be too activated, because if it's too activated, it can start attacking normal tissue. And this is an example of someone with rheumatoid arthritis, where the own immune system starts attacking the joints. And so that's an autoimmune disease. And we don't want the immune system to be overactivated, attacking the normal tissue. So it's a real balance, fighting off the enemy, but keeping ourselves well. So what are we doing in cancer? Cancer is a nasty, but it's seen as family. So your immune system does not attack it. So what we do is we activate the T cell. The T cell can be activated with these green little receptors it has on it, or it can be stopped by these red receptors. So what we do, there's many receptors, you don't need the detail, is we basically put a brake on the brakes. And that's where these drugs work. And we push the immune system to activation against cancer specifically. Sometimes we get a bit of overactivation and we get side effects, which I'll discuss. But that's the concept. It's a very specific mechanism of action. I now would like to show you the mechanism of action in cartoon form. This is melanoma here. And this is a cell that lives in our skin called an antigen presenting cell. It's very important. It's an immune cell we all have. It fights off nasties. What the antigen presenting cell does is it gobbles up the melanoma effectively, and it presents a little piece of the melanoma to our T cells. So these are both immune cells. The blue and the pink are both immune cells in our immune system. They talk to each other. And the T cell looks at that melanoma, goes, is that foreign or is that family? Is this an enemy? Is this a nasty or is this family? If the T cell is activated by this receptor called B7, it says, family. That's family. Anything that has anything like it in it, the little black bit, I'm going to just leave alone. And so the immune system sees the melanoma as family. But if we are able to inhibit that interaction, we can activate the T cell to make it kill the melanoma. And that's what Yervoy is. It's called an anti-CTLA-4. It's pink. And that's how it works. Now let's talk about anti-PD-1. This is our foundational treatment in melanoma. So what happens is our T cells, some of them do get activated to fight off melanoma. However, when they get activated and then find a whole melanoma cell, what the melanoma cell does is it puts up this defense, this PD-L1. It binds the PD-1. It's like shaking the hand of the T cell, and the T cell gets deactivated. So it's going in to kill the melanoma because it's activated, but then the melanoma throws it this signal that switches it off. So there's no melanoma cancer kill. So PD-1 interrupts that handshake, enabling our immune system, the T cells, to kill the melanoma cancer cells. So when we put them together, we activate it in two ways. We are able to, there we go, sorry, it's a bit slow. In the periphery of the immune system, at the skin level, we activate those T cells with Yervoy, ipilimumab, and then we grow an army of T cells that go right down into the tumor, and the PD-1 helps us kill the tumor. So when we put them together, we have a greater effect against the cancer, but not everybody needs this activation from the ipilimumab. Some people only need the PD-1. So that's the mechanism. So what about autoimmune side effects? Because we can overactivate those T cells, the immune system. Fatigue is very common with immunotherapy, but it does go away when the immunotherapy stops. Rashes and something called vitiligo is quite common. That's where the immune system attacks the skin. The immune system can attack the bowel and give you diarrhea and colitis. It can attack the thyroid, the pancreas, the pituitary gland. These are hormone producing glands, so that they don't work. We can replace those hormones. And then it can attack the lungs, the kidneys, the liver, the nerves. In fact, anything can be attacked, but these are much, much less common. And same with the pancreas, that is much, much less common. And it depends on what immunotherapy you are on as to your risk of a side effect. These drugs work in brain metastases. So this is a patient of mine with 3 brain melanoma deposits. And at week 18 on combination immunotherapy, he had a complete response. They completely disappeared. And I'm very happy to say that at 5 years, he remains melanoma free. So it was in Australia that we actually, with Grant McArthur and myself and our colleagues around Australia, we did the first trial showing how active immunotherapies were in brain when combined together. So this is called the ABC trial, and the yellow is the arm of the patients who got the combination, and the blue is the arm of the patients who got nivolumab. And again, 100% of patients, this time it's the relapse or progression-free survival, not the overall survival. So 100% of patients have not progressed at time 0, and in time patients progress, but by 6 months, we don't have any progression, and the remainder of the patients except 1 do not progress. And over 50% of patients have a great response with combination. And you have to remember that only 10 years ago, if you had melanoma in the brain, and if you had more than 1 or 2 melanomas in the brain, your survival was on average 16 weeks. And we now have 50% of people living or not progressing to 3 years. So this has been a real game changer. But the reason we use combination for melanoma in the brain is because it seems to be so much more active. And this was repeated in an American study of combination showing exactly the same results. So how can we solve this problem of people not responding to our drug therapies? The best way is to collect tissue. And so at Melanoma Institute Australia and other research institutes, I know at Peter Mac as well, we try very hard to collect tissue on novel therapies. We will try to biopsy before we start a drug treatment. We'll give a novel drug therapy, and then we'll try to get a biopsy early on treatment because we're trying to understand why some patients do really well and some patients don't. We then use all these different samples, so the tumor, the blood, or the feces, and analyze it to try and understand what drives response and what particularly drives resistance, because that is our greatest unmet need, resistance. I'm just going to show you one slide with a little bit of the science we've been doing. So this is a heat map, and what we've done is looked at the tumor from people who have not responded well to people who have responded very well, and we've compared the differences and tried to distinguish them. Now, blue is a good responder, orange is a poor responder. What we've done is we've taken 2 specific things that we look at in the tumor called, you don't have to worry about what it actually means, but it's interferon gamma and tumor mutation burden. And we see that patients who have high of both, so this is tumor mutation burden here, interferon here. So if you have high of both, so this quadrant here, almost all of them do well. However, these patients here who have high interferon gamma, but not so high tumor mutation, or high tumor mutation, but not so high interferon gamma, we should expect to do well, but some of them don't. They're in orange. And then on the other hand, there are some patients who have low of both, and they do well, these 4 blue dots. So what we're doing now is, rather than looking at bulk, now we've got an idea of what the bulk analysis can tell us. We're now looking at these individual discordant patients to try and understand precisely what is driving their resistance or their response in the face of what we expect to be resistance. And that way, we hope we can design more novel drug treatments for those patients who need our help. We're now at the stage of where we are thinking and doing clinical trials where we look at the patient's biomarkers, decide before we give drug therapy whether we think they're going to do well or not so well. And if they're going to do well, we give them the standard treatment. And if they're not going to do, if they're predicted not to do so well, we try to get them onto an individualized clinical trial. So the immune system's complex. I just want to summarize it in a really sort of four-step cycle so that you can understand the number of drug therapies or drugs in development out there. And we have to remember that only 4% or less of drugs in development actually get tested in humans because they just don't pass the test of activity. So in the immune system, the first thing that's important is that your immune system recognizes the cancer. And that's that blue antigen presenting cell I showed you before. After your immune system has recognized the cancer, we want your immune system to build an army of immune cells against the cancer. We then want that army to travel to the cancer and then kill the target, the cancer. Now, at the moment, we have trials of all these different drug therapies, some of which will not be successful, some of which will be successful. But you can see there's a plethora of combinations. You know, how do you decide which drug to combine with what? And in fact, the science and the clinic are neck and neck. We can't keep up with the clinic and we can't keep up with the science. So we've got to find fast ways to try and work out how best to test new drugs. So I'm now going to turn to a platform that we're using, which is proving to be very effective. First of all, just a bit of background. Adjuvant treatment is what we use in stage 3 melanoma, and that's where we resect all the melanoma we can see. And then we give drug treatment just for 12 months because we're trying to prevent stage 4 or metastatic disease. And we know by doing this, we reduce your chance of becoming stage 4 by half. It's not foolproof. It reduces the risk by half. And there are 3 drugs that are now approved in Australia. I won't go through the details, but both targeted and immune drugs for this purpose. As I said, they reduce the risk by 50%, and they're on the PBS for resected stage 3B, C, D, and 4 melanoma. So with that information, we're now using neoadjuvant therapy. So this is the exciting part in melanoma, and that's where we take stage 3 melanoma, where we know it's resectable at the beginning. We know we could cut it out, like the top line, but we give drug first and then we cut the tumor out. Then what we do is our colleagues, pathology doctors, like Professor Richard Scollier, my co-medical director, will look down the microscope at the tumor and tell us whether the patient has had a complete response pathologically, a near complete response, a partial response, or no pathological response. And all this occurs in 6 weeks. So rather than having your surgery and then having your drug and not really knowing whether you're going to recur, you sit waiting for your scan. What we're now doing in clinical trials is giving the drug first, then resecting, and then we can give feedback to the patient about whether they've responded or are sensitive to the drug. So what have we seen? Now, this is the last set of survival curves I'd like to show you. But before that, this is one of our patients. Her PET scan, you can see in her armpit, she's got a large, massive melanoma. This is her heart. That's normal. It's very active, so it takes up the sugar. But you can see after 6 weeks of treatment, it completely disappears. And this was resected. And Richard told us she'd had a complete pathological response. And good news, she's now 4 years down the track with no recurrence. And people with bulky lymph node disease like this, their risk of recurrence is up near 70%. So what are we seeing? So this is another survival curve. It's the last I'm showing you, and it's recurrence-free survival. And again, 100% of people have not recurred at the get-go when we start the neoadjuvant drug therapy at time 0. This is time on the x-axis. Now, as time goes by, you can see that these people here are not recurring. It doesn't really drop below 100%. Now, remember, in this population where we give the drug first, we expect it to drop down to about 70% by 3 years. And at 2 years, it's still up there at nearly 100%. All three groups here, the blue, the yellow, and the green, had a pathological response, whether it be complete in the yellow, near complete in the blue, or partial in the green. They're all similar within statistical analyses. And then in the orange, these are the people who did not have a pathological response, and they tend to recur quite early, but there is some group that do respond within this group and stay well long-term, and we're trying to tease them out. But the big news is that if you have any pathological response on immunotherapy, then you tend to do well long-term. For targeted therapy, we see you do well if you have a complete pathological response, but it's not quite the same as immunotherapy for all pathological responders. Whereas for targeted therapy, even if you only have a partial pathological response, it's equivalent to no pathological response. So it's only the complete responders that do well, but not quite the same as immunotherapy. So is this better than adjuvant therapy? And I just want to quickly show you the importance of randomized trials. This is only a small one of 10 patients in each arm. So we call this a pilot study. It's not a study that will help us get drugs funded around the world. It's just a signal. And in this signal finding study, we see the relapse-free survival and the overall survival appearing to do better for neoadjuvant than adjuvant. So now what we're doing are large randomized trials to prove that it works. This is not enough proof. 10 patients in each arm is not enough. And so there are trials underway to look at that question. So as I said before, a pathological response is great because we can feed back to people after simply 6 weeks of drug. And if they haven't had such a good response, we can alter their follow-up, either add in radiotherapy or change to targeted drugs if they're on immune drugs, etc. So this platform's marvelous because now, so this is the neoadjuvant where you give drug first, then the surgery in stage 3. We can test drugs quickly in 6 weeks. We now have benchmarks for our standard drugs, standard immunotherapy and targeted therapy, but we can quickly test novel treatments, novel combinations. These are all trials we have running right now to see whether they do better than our current standards and whether we need to take them into bigger trials. So we do have many trials underway. I'm just going to finish off on this. Trials are the only way to push that 50% higher. And I've put here at the top the different stages to remind you of melanoma. So in stage 2, resected melanoma, we have an adjuvant trial of a PD-1 drug versus standard follow-up, which is no drug. That is standard in stage 2. In stage 3, resected, we have personalized vaccine plus PD-1 versus PD-1 alone, which is standard at the moment. And in stage 3, we have a pegylated version of another immune therapy plus PD-1 versus our standard PD-1 alone. For neoadjuvant, we have 3 trials open at the moment of various different novel combinations. And for stage 4, for patients who have never had any treatment, in stage 4, we have 8 trials open, 2 of which are brain trials. And for stage 4 patients who've progressed on anti-PD-1, we have 6 trials. Now, to be fair, it is better if we can get our patients to be seen at large melanoma centers at this point. It is easier to manage melanoma in the setting of all these different clinical trials early on rather than at progression. But if patients do come to us with progression, we do have clinical trials exploring novel agents in that setting. And all the trials can be found on this website, clinicaltrials.gov. You simply put in your condition or the disease or other search terms, your country, and all the trials pop up. But you have to make sure that a patient is eligible because there's strict criteria as to who the trials suit. So I've covered the standard therapy in melanoma as it stands in 2020. BRAF targeted therapy and immunotherapy. I've talked about the mechanism of action, the side effects. I've talked about how we're trying to improve the outcomes in melanoma by collecting tissue, the neoadjuvant therapy, and clinical trials. So with that, I'd like to thank, importantly, all our patients and our families who are participating in research for themselves and for the future generations, and all of my colleagues around the world, and particularly those at Melanoma Institute. Thank you. I'm happy to take any questions if there are some questions. I don't know how much time we have, Victoria.
Thanks, Georgina. That was a fantastic presentation. So much information to take in. One question we had sent to us this afternoon was about how common is colitis with the immunotherapies as a side effect?
It all depends.
And it's a two-part question, I have to warn you.
I'll do the first bit. So for PD-1 monotherapy, so just a single drug like pembrolizumab or nivolumab, it's about 15%, and it tends to be low grade, meaning not very severe. For about less than 1% will get a very severe diarrhea. For combination immunotherapy, so when we combine the PD-1 with ipilimumab, Yervoy, it's about 40%, and it tends to be severe. Most patients we can treat with switching off the immune system for a period of time to bring it under control, but there are some patients who are refractory to even that treatment, and we have to increase the immune switch off, or what we call the immune suppression, to get it under control. And at the very worst, sometimes we have to operate on the bowel and remove it, but that is rare, particularly in our hands where we have so much experience in Australia now with these immunotherapies, but occasionally that can happen.
Okay, so if you were to have colitis and have some steroids and then things settled down, would you be able to continue on then with immunotherapy?
It depends on a number of factors. It depends on how severe it was, what drugs you're on, can you continue on one drug, what your disease is doing, can dosing be modified? It all depends. There's not a single answer to that. Yeah.
Okay. Another question that's come up is with neoadjuvant therapy, and I know that's very new, so it's not widely understood. If you have a palpable lymph node and you have your 2 doses of immunotherapy or targeted therapy, whatever your doctor has prescribed, and then you go and have surgery, is it a complete dissection, or do they just look at the palpable node to see if it's gone down?
That's a great question, and I didn't show the data on the Prado trial. Current standard for a palpable node, this is the standard across the world, would be upfront completion lymph node dissection. That's the standard. If you're on a neoadjuvant trial, neoadjuvant is not standard of care, it is only in trials, then you get your drug and you'd have a complete lymph node dissection. So you remove all the lymph nodes. However, there is one clinical trial we are doing where we identify at the get-go the largest node, and we call that the index node. We remove only the index node at week 6. Richard looks under the microscope and tells us if they've had a complete or near complete response, or if not. And if they've had a complete or near complete response, they then do not have more surgery. They only have the one node removed. If they've had anything less than a near complete response, they get a completion lymph node dissection. But that is only in clinical trials. Okay. I hope that answers that question.
Yeah, thank you. Now I'm getting quite a few questions about subcutaneous melanomas like uveal and acral, and they relate to whether immunotherapy works with them.
So I didn't cover that. I mentioned that at the beginning, that this was mainly the data was about skin, cutaneous, which is the most common. So acral is actually a skin melanoma. It's just on the thick parts of your palms and your soles. It does respond to immunotherapy, but less frequently than cutaneous melanoma. So the response rate is lower, but some people can respond to immunotherapy. And it's about 2/3 of the activity in skin occurs in acral. For mucosal melanoma, that's melanoma that arises on the inside linings of the body, like inside the mouth or the gastrointestinal tract. And they again can respond to immunotherapy, but again, it's not as active as in skin. It's about half as active, but people still can respond. And mucosal melanoma is one where we try to use dual immune therapy together because it's more active than just one immunotherapy for mucosal melanoma. It's actually a huge area of our research, trying to find treatments that can activate the immune system against mucosal melanoma. So that's those two. Then uveal melanoma is different again. It's a completely different tumor. It's called melanoma, but it really is very different in terms of its pathology. It metastasizes or spreads often to the liver. It is not responsive to immunotherapy. I think the rate for dual immunotherapy is around the 10% mark, but I must say in the real world, it's less than that. We're not seeing a lot of responses. Yeah.
Okay.
And we're
Thank
working
you.
on that as well.
Now, another question I have is about if I have stage 3 and I've had immunotherapy, and let's say I'm a woman, well I am, but and I'd like to have a family. How soon after having immunotherapy can I think about having a baby? Should I be thinking about it at all? And how many years should I wait?
They're all excellent questions, and immunotherapy has changed what we would have said 10 years ago. So 10 years ago, it was often advised by the medical team that if you've had stage 3, particularly if it's high risk stage 3, that you really should not consider a family for at least 5 years. And some physicians advised not to have a family. However, things have changed in many ways, not only our drug therapies, but the realization that it's an individual decision. So we don't have any strict rules about it, but the most important thing is that in any discussion about having a baby or getting pregnant, that several things are discussed and the couple completely understand. Number one, what is the risk of recurrence, even if you've had treatment? And the recognition that if you have a recurrence, and this is hard to say, but your life may be shortened, and has the couple considered what that would be like for their family if one of the parents died? So that just has to be discussed and understood. The risk may not be high, but it has to be discussed so the couple can discuss what they'd like to do. The second risk is what is the risk of hormones in pregnancy on recurrence? And that should be discussed. We don't have a lot of good data on that. And then the third is, should you recur while pregnant? How can that be monitored and managed? So it's with that context that a couple will discuss with their team when they may want to have a child and the timing of that. Generally though, I advise my patients that we don't really have a full-on discussion about all the ins and outs until at least 2 to 3 years after surgery. We also advise if you're going to have a baby that you have at least 6 months off drug therapy. We don't think that immunotherapies affect the fetus, but we don't really know. So we advise a washout of 6 months. But the bottom line is there are no rules about whether or whether not to have a child. It depends on the couple and the discussion about the risks and benefits. And we go from there, basically. I hope that answers that question, but there are no rules.
Very thorough. I have one question, and this comes up time and time again. This person is on immunotherapy, and is there a recommended diet or vitamins to be taking while you're on this type of therapy?
There are no recommended dietary requirements. So the things I say to my patients is, number one, diet is complex. We don't exactly know specific aspects needed, and everything should be in moderation. We don't go down one channel. That's not healthy in anything, to be doing only one type of food. We know in health in general and in cancer prevention that a diet which is broad, that follows the seasons, that has a lot of vegetables, legumes, and fruit, but not more vegetables than fruit. Anything that is good for the microbiome, which is broad, is what I recommend. Plus, I give a general principle. I offer for the patient to go and see a dietitian, but the general principle is if you're producing a lot of rubbish in your kitchen bin, then you're probably eating not healthy food. So packaged stuff really should be limited to things like rice, legumes, and pasta, not biscuits, etc. But all the things we tell every human being about their diet. But there's nothing specific we say. I personally think vitamins are an expensive way to make expensive urine, because most of it, if you don't need it, you will pee out. And if you've got a good diet, as I just mentioned, then you tend not to need those vitamins. They will just be peed out. So we don't tend to advise specific probiotics either, because we don't want the gut to see only one sort of nutrient. We want a breadth. And believe it or not, passing wind means you're probably eating the right stuff, lots of beans, and that's good for you.
Okay, good. What about, dare I mention the complementary therapies topic? That's very wide ranging, I know. I don't know whether you've got time to answer that one.
I think it's a big topic, but needless to say, we know that 70 to 80% of our patients are seeking alternative medications, naturopaths, various other treatments. The only thing on clinical trials, it's just not on. It's too dangerous, actually. The only thing we say, and I always talk with my patients about their alternative medications if they're on them, I'm very happy to, is we don't have the same level or bar that they're kept to. So you heard me talk about randomized trials and the need to prove things. None of that applies to alternative medications. They don't need to reach that bar. They're approved with very little evidence. But in the medical world, we have to spend a lot of time doing these clinical trials to prove their worth. So what that means is the level of evidence is low that they're effective, and the level of information on their toxicity and negative effects is very low as well. And that can be dangerous. I have several patients who've used lots of turmeric, for example, and I've had to take them off their immunotherapy because their liver's tests became very abnormal simply at the time of starting the turmeric. When they stopped the turmeric, things improved and I was able to relook at it. But high doses of alternative medications can be dangerous, and we need evidence as to their effect being positive and understanding their negative effects, and we don't have that data. Just because it's natural, it doesn't mean it's good.
So true. All right, Georgina. Well, I think that's about all for the night. Is there anything else you'd like to add to your presentation?
No, I'd just like to thank you for the invitation, and it's a wonderful venture to be able to talk to our patients in this way.
Thank you.
No worries.
Very appreciative.
Thank you.
All right. Thank you. Wow, that was a good presentation. So now I'll move on to our very special patient speaker, Narelle Wish. Narelle has been a long-time supporter of MPA and regularly comes along to our Melbourne support group meetings. In 2009, Narelle had a small melanoma removed from her leg, but unfortunately in 2016, it came back via a palpable lymph node in her groin, and she had a full dissection. Now, following this surgery, she decided to go on to one of the new adjuvant therapy clinical trials that was around, which has proved to be a very positive decision for her. Tonight, she's going to share her personal experiences of living with melanoma and the ups and downs of being on a clinical trial. A very warm welcome, Narelle.
Thank you. Thank you, Alison, for the introduction and for asking me to share my experience of being on a clinical trial. I feel very honoured to be in the company of Professor Grant McArthur and Professor Georgina Long tonight, both whom I hold in high esteem. My journey with melanoma began when I was 39 in March 2009, and my husband noticed a suspicious looking mole on the back of my left leg just above my knee. I attended my GP and she was instantly concerned, even making the appointment herself for me to see a dermatologist. You would think her concern should have triggered some kind of concern in me, but it didn't. I attended the appointment and what ensued was a diagnosis of stage 1 melanoma. I had a wide excision done, and once it had healed, and because it wasn't visible to me, I put it to the back of my mind. I went regularly to my dermatologist for continuing skin checkups with no new spots popping up. Fast forward 7 years to February 2016, and melanoma came back with aggression. This time I had an annoying lump in my left groin that just wouldn't go away. It wasn't causing me any discomfort until one weekend when my whole left thigh area became red and quite sore. I spent the weekend icing it, and by the Monday it was looking a lot better. At the insistence of my husband, I went and saw my GP on the Monday. What followed in the next few days were more medical appointments with ultrasound, biopsy, CT scan, and finally a stage 3B melanoma diagnosis. Something that shocked me and my GP. I did some research on my treatment options and was quite uncomfortable that the only options at that time were removal of the tumour, possible radiotherapy, then a watch and wait scenario. Once the family and I had digested the news, it was time for action. I had a full groin dissection in March 2016 with 24 lymph nodes removed and one testing positive for metastatic melanoma. During the surgery, some of the nerves in my thigh were cut to remove the nodes, so I have lost a lot of sensation and feeling in my thigh. After surgery and in consultation with my oncologist and surgeon, I was given 3 options for further treatment. Option 1 was to watch and wait. This option did not sit well with me at all. Option 2 was a course of radiotherapy. Again, not my favourite option as I was concerned about lymphedema, and radiotherapy gave me a greater chance of this happening. Option 3 was to participate in a clinical trial. This was by far my favourite option. I did extensive research into clinical trials, spoke to my family and reached out to Alison. As I have twin daughters who at the time had just commenced year 12, I wanted to do whatever I could for them and me to make sure I had the best opportunity to be around for a long time. So I opted for the clinical trial. The trial I was offered was adjuvant immunotherapy with Keytruda versus placebo after complete resection of high-risk stage 3 melanoma. It was a double-blind phase 3 trial with a 1 out of 2 chance, so a 50% chance of receiving the treatment drug. There were side effects that also needed to be considered. After much thought, I registered to do the clinical trial. I felt it was a wonderful opportunity to participate in such important research. I knew there was a chance of getting the placebo drug, but I believed it was still worth participating. I would be closely watched, and should the disease come back during my treatment, I was already in the right place to receive the treatment I required. The trial treatment was going to include 3 weekly infusions, blood tests, and doctor's review. Every 3 months, I would be given a CT scan. I started my treatment trial at Box Hill Hospital in May 2016, about 8 weeks after my full groin dissection. The treatment ran for 1 year with an infusion every 3 weeks, a total of 18 infusions. I had CT scans every 3 months, wonderful support from the nursing staff and mental health assessments. I was so closely monitored that I knew if I had a recurrence, I would be well looked after. My side effects included tiredness, lack of concentration, heartburn, itchy back, loss of skin colour, lack of energy, and upset stomach at times. However, these were all manageable. I finished my clinical trial in May 2017 and am now having 6 monthly CT scans and visits to the oncology team at Box Hill Hospital. My scans continue to be clear. It is now 4 years, 7 months since my stage 2 melanoma diagnosis and full groin dissection, and 3 years, 5 months since I had my last clinical trial treatment. Once I reach 5 years post-diagnosis, which is next year, I have an option to be discharged from the clinical trial or continue with 6 monthly CT scans and oncology follow-ups. I will be staying on the trial for as long as I can for my own well-being and peace of mind, and also to assist with the ongoing collection of data for the clinical trial. I'm so thankful for the worldwide research into melanoma and grateful for the opportunity to be part of and assist with the research by participating in a clinical trial. Being diagnosed with stage 3 melanoma has changed my life in many ways. I have mild lymphedema in my left leg, so wear a compression stocking daily. This and my scars give me a visual reminder of what I have gone through every day. I changed my work life to a more balanced one and cherish even more every moment I get to spend with my daughters, husband, family, and friends. I've met some wonderful people on this journey of melanoma who have guided and supported me and who have now become friends. Being on a clinical trial for me was the best option I could have taken.
Thank you, Narelle. That was a very insightful story. And, you know, I do think stories such as yours do give hope to others. And I'm sure many here tonight will appreciate the impact that melanoma has had on your life. It will resonate with them as well. So I thank you very much for coming along tonight.
You're welcome. Thank you for having me.
Pleasure.
Thank
you. Okay, so now I'd like to introduce our next guest speaker, Professor Grant McArthur. Last but certainly not least, Professor Grant McArthur is a Fellow of the Royal Australasian College of Physicians and holds a PhD in medical biology. He is the Executive Director of the Victorian Comprehensive Cancer Centre. He is the inaugural Lorenzo Galli Chair of Melanoma and Skin Cancers at the University of Melbourne and is a Senior Principal Research Fellow with the National Health and Medical Research Council. Prof McArthur is also head of the Molecular Oncology Laboratory and of the Cancer Therapeutics Program, Cancer Research. He is a Senior Consultant Medical Oncologist in Cancer Medicine at the Peter MacCallum Cancer Centre. He is a national and international study co-chair of a number of clinical trials of targeted therapies. Now, Grant's research interests include the discovery of novel drug targets in cancer, targeting oncogenes, the immunological effect of targeted therapies, clinical trials of targeted therapeutics, personalised medicine, melanoma, cell cycle control, metabolism, and protein synthesis in cancer. Grant also sits on MPA's medical advisory panel, and I would like to add that Grant has been a wonderful supporter of MPA's patient information forums over the years. And I think this is your 5th or 6th Grant with us, but anyway, welcome.
Thanks. Thank you very much, Alison, and thank you to Victoria and the whole MPA team and all of you who have joined us this evening to hear about melanoma. Now I'm going to share my screen with you so you can and go through some slides just as Georgina did, and just bear with me for a moment while I get that up. Okay, so so I think we've heard 2 wonderful presentations already this evening. You know, Narelle, you're so generous in going onto a clinical trial, and you know, I think you showed us how patients get really great care when they're enrolled on a clinical trial, and of course for you a great outcome, and thanks so much for staying on the trial and allowing all that follow-up that's going to help so many patients in the future. And really we heard from Georgina just how important clinical trials are if we're going to do even better from melanoma. We will not rest with 50% of patients with advanced disease now we think are becoming long-term survivors with our best available data, but we won't rest and we need to keep doing more research and more clinical trials. So what I want to do for you tonight is sort of do a deep dive into clinical trials, explain what they are, how we run them, some examples, couple of little controversies that might make for discussion points. So these are my disclosures that I've been a principal investigator on clinical trials in the pharmaceutical industry, and the funds from that or get paid to the Peter Mac to reimburse for trial costs. Okay, so let's get into clinical trials and what they are. Well, really, clinical trials are how advances in medicine and healthcare are tried, tested, and proven. You've heard a lot from Georgina about examples of treatment-related clinical trials, but there are clinical trials to look at new ways of diagnosing cancer, screening for cancer, and prevention of cancer. So there's a whole variety of different clinical trials. They're absolutely vital for how we take new information and implement it into practice. I'm going to show you in a moment on the next slide, trial participants do better. So Narelle, I reckon you made a great call going on to a clinical trial. And for the health system, clinical trials are a quality indicator. What do I mean by quality indicator? Well, clinical trials set standards of care, the way patients should be cared for, and so set the quality standards in our health system. Now, I mentioned that clinical trial participants do better, and this is the data of that very large set of data, almost 8,000 patients. And you can see that this is one of those survival curves down from 100% follow up through to 10 years on that x-axis. And in fact, the survival's better for patients with cancer that went on a clinical trial than those that did not. Now, that doesn't necessarily mean that it was the trial that made people live longer, but because it's not randomized whether someone went on to a trial or not. However, what we can absolutely say is that there is no evidence that going on to a trial that you're a guinea pig and you're going to do any worse. In fact, you seem to do better. So how do we do our clinical trials? Well, people like, excuse me, Georgina will write a detailed protocol that says why you're doing the trial, how many participants or people are needed to go on that trial, what is the eligibility. And eligibility is quite important because not everyone is eligible for every clinical trial. There are details required there. The protocol says what treatments will be used and how the information will be collected and reported. Now, the one thing that's really critical in the design of clinical trials is statistics. And we talk about the power of a trial. And the power of a trial is that we need enough patients so that we really know we're confident of the results. For those technically minded in statistics, reject the null hypothesis. But it's really important not to have too many patients that might slow down the development of a new treatment and get it to the general patient community around the world, or expose too many patients to a treatment that really isn't working well. And so you've got to get the numbers of participants just right for a clinical trial. So after the protocol's written, including all the statistical design, ethics committee reviews it to ensure patient safety and the scientific validity of the trial. And then there's a process of informed consent where patients are invited to participate. So if after careful consideration, Narelle gave us a nice story as to how she had a good think about the trial before she went on. But if someone decides to sign up for the trial, then they have a treatment plan, which is very protocol driven and detailed. And when the number of pre-planned patients have been treated with a follow-up time of a certain amount or a number of events occurring, that is things like, have the tumours grown or not grown, then the results are analysed. Now, in the analysis, we often use what's called a data safety monitoring committee, and they keep an eye on the trial to make sure, an independent group, that the trial is not being dangerous to patients at all, that safety is being monitored, and also when is the right time to stop it. Because, and I'll show you an example shortly of how a data safety monitoring committee was really important to get a trial finished in time for, what was then a new breakthrough treatment. Now, I'm going to get into a bit more technical detail now and talk about how we develop new treatments for cancer, including melanoma. So we do work in discovery of new scientific phenomena, and Georgina touched on these. We do test tube tests in the lab, and then we also use animal studies that are required by the regulatory agencies if a treatment is to go into patients. Then we have three classical phases of clinical trials. There's phase one, and in phase one, you work out what is the right dose, how often do you give the treatment, and is it safe? You then, in phase two, go to, well, does it actually work? Is there some evidence that this can work and be beneficial for cancer? And then in phase three, you compare to the previous standard treatment and see if it's better. And then if everything goes well, it moves routinely into clinical practice. Now I'm going to go now into a deeper dive into what we do in the different phases of trials. So in the phase one trial, we're aiming to, is the treatment safe? And can we determine the right dose and how often to give the treatment to go to the next phase of trials? We look at what's called target modulation. And this is, what does that mean? Well, it means that patients are invited to contribute biopsies of the tissue of their cancer for analysis to see whether the treatment scientifically is turning off the target and doing what it should if it's going to work. This is important because if the treatment is not turning off what you think it is, if there was a PD-1 treatment and it wasn't inhibiting PD-1, well then, we wouldn't want to pursue it if it wasn't working. We also do what's called pharmacokinetics, which determines how long the treatment, the drug, will last in the system. I remember a sad situation with what was thought to be a huge breakthrough in cancer back now 25 years ago, a treatment called an anti-angiogenesis treatment, hailed as the biggest breakthrough, it's going to cure cancer. Well, unfortunately, the treatment only hung around the body for about 2 hours, and it actually got to phase 3 before that was worked out. And so you've got to know how long the treatment works in the body for early in the treatment. Now, I'll show you now about an example of target modulation. So when we do a phase 1 trial, we often start at a low dose and then we increase the dose. And what we do is you increase the dose, we look to see if it's turning off the target. So this sort of black lines here are an example of turning off what we call a protein kinase. That's what BRAF is. And you can see as you give a higher dose, then the black lines become more faint. And that means you're turning the target off. And that's why those biopsies are so important to understand how the treatments work. So if all that goes well, you go into phase 2 trials. Now you're talking about, does it work, efficacy? And the sort of things we measure response rate. So what proportion of tumours will shrink, not just shrink a little bit, but we have a standard definition used in trials all around the world, the rate at which there's a greater than 30% reduction in the size of the tumours. Now that's very important because that means that, when Georgina and I are talking to colleagues in Italy and Poland and Canada, we're all using the same criteria when we report shrinkage rates of tumours. We also often measure time to progression. So obviously we like cancer to never progress, it'll shrink and never grow again. And that's the ideal scenario, but we often measure the time to progression, for melanoma and cancers. Georgina talked about predictive markers. Can we work out which patients are going to get most benefit? And we really do start to do that. Do a bit of it in phase 1 trials, but phase 2 especially. And again, biopsy is so important. So, I'd encourage anyone that's being enrolled in a trial, if it is possible to do biopsies and you're happy to do that, thank you so much, because it's so informative to understand the science of our treatments. And we keep measuring safety as well throughout. At the moment, there's lots of controversy about COVID vaccines and will they be safe? And President Trump's trying to rush them through. Safety is so important. It's important for melanoma treatments and super important for things like vaccines for COVID, which we hope is coming very soon. So if phase 2 goes well, the drug hangs around for a while, it turns off its target, it's causing shrinkage of the melanomas. Well, then we go to phase 3 trial. Now we're comparing it to a standard treatment. And now we start to measure things like how long do people live on the two arms of the trial? What is the time to progression? If it's a better treatment, that time should be much longer than the standard treatment. And we do measure quality of life because we want to know that people are feeling better with the treatment. And it's interesting that in melanoma, if you get tumour shrinkage, you nearly always, not surprisingly, have a better quality of life. So, but we do measure that formally. We keep trying to understand which patients are going to get most benefit, and we keep measuring safety for the reasons that I've said. So who does these trials and who? There's the investigators, Georgina and myself and others, and we have teams that we work with to run the trials. But trials are often sponsored or coordinated and have legal responsibilities for the trials by pharmaceutical companies. These are all the companies that have current approved drugs in Australia and are doing trials in Australia. So it can be pharmaceutical companies, but we also have what we call cooperative trials groups, and we have one for melanoma and skin cancer in Australia called Melanoma and Skin Cancer Trials. And these are groups of investigators with patient input and other professionals that run clinical trials, answering different questions to the pharmaceutical companies, things that we think are very important for the community. For example, the Melanoma and Skin Cancer Trials group at the moment have started a trial to ask how long do you need to stay on PD-1 treatment if you've had a response. Now that's something which the pharmaceutical industry are not particularly interested in, but we are very interested in, so you answer that sort of question in a cooperative group trial. All right, now I'm going to give you an example of a trial, and I'm going to pick something, a bit controversial to, get you all thinking, and you might pick it up in questions. So a number of years ago, the very first BRAF inhibitor that came along was a treatment called vemurafenib. And I co-led this trial with some colleagues, a colleague from the United States, colleague from Europe. And for patients that had a BRAF mutation, they were randomized. So in a trial, you can have the electronic toss of a coin. You either got the new BRAF inhibitor or you got chemotherapy. And you heard from Georgina, we don't really use chemotherapy anymore. Now, as this trial started, the results from the phase 2 trial were maturing and it started looking really good. And so this was a breakthrough treatment at the time. There were a series of articles in the New York Times all about this treatment. But then the New York Times ran an article about this randomized trial. And when testing a drug means withholding it. So here were two cousins, front page of New York Times. One got the BRAF inhibitor and one got the chemo. And is this fair? Is this the right thing to do with a new treatment? There were articles in the New England Journal of Medicine, which is the top medical journal in the world, about all the ethics of this. Now, as leaders of the trial, as this new information came in about the treatment looking really good, we felt, yes, there's a case here to try to get this trial done and finished as soon as we can. We decided not to stop it. We thought it is actually ethically important to continue it because without a randomized trial, it was not going to get approval in places like Australia or Europe. So we did a new statistical plan, and this is lots of technical data, but statistics are so important. But what I've highlighted in the box there is that we changed the statistical plan so the trial would be finished when there was only 200 patients across the whole trial sadly passed on, rather than initially the plan of 400. And we thought that was the ethical and appropriate thing to do for the benefits of the whole community, plus of course the people on the trial. Interestingly, I had one patient who got the chemo on the trial and had a great response, who, when I rang her to tell her the results of the trial and would you like to change over and have the new BRAF inhibitor, said, no, I'm doing very fine, thank you, I'll stick with the chemo. But that's what happens with a trial. The Data Safety Monitoring Committee say, right, you can now finish it and tell the patients to give them the opportunity of changing over to the other treatment. Okay, so I've shown you this before. This is that phase 1, phase 2, phase 3. Now that can take a long time. And is it the right thing to do if you've got a really breakthrough treatment that is obviously working well? So is this paradigm of having to go through the phases changing? Well, it did change with Keytruda, the drug that Narelle received. So this is what we call a waterfall plot. So what's that mean? So what it is each of the coloured lines below the white horizontal line is patients that if it goes below the line, it means the tumours have shrunk by a certain percentage. So some of those patients have shrunk 100% or complete response. And then some other patients did not respond to Keytruda and there was growth of the tumour. But overall, 72% of patients had tumour shrinkage. That's pretty impressive. Not only that, but when the trial analysed the survival of the patients that received Keytruda, the average survival for advanced melanoma before the BRAF and the Keytruda came along was only about 9 months. But you can see here that about 50% of patients were alive at 2 years. This was obviously better. And so based just on the phase 1 trial, without going to phase 2 and 3, there was approval of Keytruda and access. And Australia was actually the first jurisdiction in the world to give approval to Keytruda in the first line, that is before patients had the other drug Yervoy that Georgina also spoke about. All right, so that's a summary of how we do trials, and things are changing, right? We're starting to try to make going from the first phase to getting the drug approved much quicker. Now, what I want to do just for the remaining time now is talk to you about other innovations that are happening in clinical trials, new ways of doing clinical trials. So one thing we're doing is starting to do what we call a registry trial. Now, what's a registry trial? Well, what we have across many areas of healthcare is registries of clinical information that record how patients are doing. They're generally simple. They don't have lots and lots of details like we collect on formal clinical trials, but they have the most important information. What's the diagnosis? What are the most important parameters? And what's the outcome for those patients? So a number of years ago, a Swedish group did a registry trial where they had this registry that covered all the hospitals in Sweden. And on this registry, patients were invited to go onto a registry trial, which was to determine if they'd had a heart attack, whether sucking out the blood clots from the coronary arteries to the heart would improve outcome. So an amazing 7,000 patients got randomized, which actually was about 60% of all the patients in the time period of the trial in Sweden that had a heart attack. So that's pretty amazing. You know, in cancer in Australia, you know, at the moment, unfortunately, it's less than 10% of cancer patients get on a clinical trial. They got 60% of all patients that had a heart attack on a trial. And they did the trial very quickly in less than 3 years. And what is amazing, $50 a patient to get the answer. And the answer was that sucking out the blood clot didn't matter. But this is a pretty interesting new way. And people are talking about this being the next disruptive technology. There's some other advantages. I mentioned the cost, but there's other advantages. So in the sort of trials like the BRAF trial I showed and the immune trials that Georgina's speaking about, the eligibility often gets patients that have quite similar characteristics. It's easy to analyse the data that way. Where in a registry trial, you can do that in the real world and you have all the variation that happens between patients in the real world and determine how the treatment does in a real world setting. So the registries where we collect clinical information, they're real world, so they're typical patients. They're not highly selected patients for trials. They're quite low cost information to collect. The data quality is not as deep as you get on a trial. In a typical randomized trial like that BRAF trial I spoke about, the randomization gives you the advantage of of internal validity. You can really be confident of the outcome if one treatment's better than another, but they're really expensive. You know, those phase 3 cancer trials can cost hundreds of millions of dollars to perform. They're complex, and that adds costs ultimately to the cost of drugs. So what we're looking at doing now, and have started actually, is these registry trials in cancer where you do that randomisation on the registry. And these are some of the centres around Australia that are participating in registries where these registry trials can be applied. We haven't got one in melanoma yet, but coming, and we think this will give a lot greater access of patients to clinical trials. The next area of innovation I want to talk about is called teletrials, which are really interesting way to improve equity of access, particularly patients in regional areas and other underserved patient groups. So the idea of teletrials is to really open up the opportunity of people, regardless of where they live, to be able to get onto a clinical trial. And all the benefits that we've spoken about of being on a trial. So the idea of it is that you have a primary site and then you have satellite sites. So for example, you know, one of the things we've been running, there's a Peter Mac is a primary site for a teletrial, and some of the regional oncology centres in Victoria are satellites. And at the primary site, there's a lot of detailed information collected, where you just receive the treatment, and things are simpler at the satellite sites for those sites they don't have to do as much of the work for the trials. So why are we thinking about doing this? Well, actually, there's a lot of trials that a lot of sites that don't recruit many patients. A lot of trials don't keep patients on the trial. Thank you, Narelle, you're staying on the trial. That's wonderful, but retaining patients on trials can be a challenge, particularly if you live a long way away from the trial centre. Trials usually take longer to complete than we would like, and we think with this teletrials that can be all sped up. So increase equity for people that live in regional and rural areas. If you've got a rare cancer, it's very hard to open up a trial at every single hospital in the country, but via teletrial model, potentially, you know, the vision is that you could do that. We love the networking between places like MIA with Georgina and smaller centres, and that's the vision there, that will get the really networked care of patients firmed up. And it also helps develop, because of the quality aspects of being involved in trials, the quality of care in regional areas. Now, finally, in finishing up, I just want to talk about finding your way to a clinical trial, and how you can find clinical trials. So there are a variety of websites available that can help you find trials. So this is one that's in Victoria, the Victorian Cancer Trials link, and there's an app, and you can put information here and find trials. Georgina mentioned before clinicaltrials.gov, which is the US site. Now, this is actually fantastic. This is where I go to look for trials, but it's very user-friendly, and patients, supporters and carers can help out with this as well. There are, believe it or not, 355,000 trials listed in 217 countries. And great that the US government are putting this up. So we put all of our trials on that here in Australia. So it's actually quite a good way of finding trials. A lot of information, of course, but believe it or not, with the search, if you put melanoma in Australia, you know, you'll get a list of trials. The Australian government also has a trials app, a website that you can search for as well. There are some limitations, however, to these websites, in that the information is not always absolutely contemporary, because it can take a while for something to get listed, and then when a trial closes, it takes a while for that to come off the website. We haven't quite perfected the efficiency of that yet. So the other way of finding out about clinical trials is your oncologist, like the great Georgina Long. But all oncologists nowadays are trained in trials, are trials aware. They're keen for people to consider clinical trials, and you can ask your oncologist. Look yourself, yes, ask your oncologist. And I think every patient with melanoma should ask their treating clinician, or are there any trials available for me, because you get such great care. Now, just one final thing before we break for the Q&A. And I put this slide in even before we had questions for Georgina, because I thought you guys would be really interested. So at the VCCC, we're doing a lot of webinars like we're doing this evening. Now, Monday week, the 26th, 1 p.m, that's Eastern Daylight Savings Time. I'm really happy to have the amazing Dr. Jennifer McQuaid, who's from the MD Anderson Cancer Centre and is an expert in diet, gut microbiome, and energy balance on outcomes for immunotherapy. I reckon you guys would, she gives a brilliant seminar and you'd really enjoy this. So if you search in Google, VCCC Monday Lunch Live, you'll find your way and you can register for that. And please feel free to join us. So thank you very much for your attention. Look forward to the discussion. Thank you.
Thanks, Grant. That was quite an in-depth presentation of clinical trials. You really don't, when you go on one, I think the first glance that people have about the complexity of clinical trials is when they get given all that paperwork to read. And I'd like to know, I've had many patients ask, why am I given all of this paperwork? Some have said, I felt so scared, I don't want to sign on the dotted line now. Yeah. So I'm just wondering if you could explain why it has to be so thorough.
Yeah, no, look, it's a very important question, Alison. So, things have changed dramatically over the last 30 or 40 years. You know, back in the 1980s, consent form for clinical trials, 2 pages, and now they can be 30 pages of information. And unfortunately, because of the perceived legal risks and the need to give all the information that's possible in that information form, that's why they're so long and complicated. What my advice is, you ask your doctor to, summarize what's important for you. So you've got all the information, but what's important. And actually, most patients don't fully read them. Most patients actually will, trust their doctor, but we do encourage people to read them, because when you consent for a clinical trial, it should be truly informed. So you should read it, but we know that actually many patients don't. You can ask your doctor, you can ask your study coordinator or research nurse some questions. And occasionally, I do get people to talk to their GP, or even sometimes get a second opinion about the trial if they're not sure. So yeah, it's unfortunately is a complicated process, and there's no simple way of making that easier, unfortunately.
I was going to ask you about teletrials, but you've covered that so well, because of the people in rural and regional areas. And I'm wondering, is this just something that's been used now in Victoria, or is it will it be around Australia with the major hospitals going into hubs?
Yeah. So look, this is I think a very exciting development, and we were delighted that the Medical Research Future Fund has just funded to the level of $75 million, a national program to roll out teletrials across the whole country. It's not going to happen overnight, but that's a fantastic commitment. And my colleague, Dr. Sabe Sabasan from Townsville, is leading that program nationally. He's real national leader in teletrials. So Queensland did the first teletrials in Australia. We're now doing them in Victoria, and I would think over the next 2 to 5 years, we'll see a very big expansion. I think it's very exciting. I have this actual dream that we could have virtually every melanoma trial and other cancer trial available to everyone in Australia through teletrials without the need to travel. Now that is probably a bit optimistic, but nonetheless, I think that's what we've got to aim for: accessibility. Not every trial can you go on no matter where you are. If you have to have a biopsy or something, that can't be done at every site for the trial, but I think for some trials we can open up very, very widely and have treatment close to home and not have to go to the city if you live in a regional or rural area.
So with the effect of COVID on clinical trials, have any of them stopped or been paused rather? And are there less available at the moment because of COVID?
Yeah, now there's, COVID has been, such a difficulty for everyone in so many ways, of course. And that included impact on clinical trials. So very early on, when we were quite worried that Australia might go the way of Italy and Spain and have huge outbreaks, and health systems where our intensive care units are overflowing, we took quite some conservative steps to slow down clinical trial recruitments. But what we learnt is that, we've learnt to adjust to the challenges of COVID, and now most units have all their trials open. So they were slowed down for a while, and that was difficult. But now, we've learnt, despite, the difficulties, and here in Victoria, we've had the greatest challenges with COVID, but pretty much now back up to full speed for trials.
Sure. Okay. Now I do have, and probably this will be our last question, if that's all right. Is gosh, what was it? It's gone. I've had a mind blank, Grant. Help. Oh dear. Liquid biopsies in trials, are they becoming more common?
Yeah. So liquid biopsies, some of the people listening will know exactly what they are, but the idea of the liquid biopsy is to take blood and then analyse features that reflect the cancer, the melanoma, in the blood. So what's really interesting is that the DNA inside the melanoma cells leaks out into the blood, and you can measure that in the blood and see what is happening to the DNA coming out of the tumour. Not only that, but research now, with all this interest in things like microbiome, you can find what type of bacteria you have in your bowel from doing a blood test. Pretty amazing stuff, really. So the idea that you can extract, a lot of detailed information from a simple blood test, rather than having to, in every case, do a biopsy, is a really exciting technology. You can, of course, go back and measure blood at multiple points in time. And so, where that technology is at the moment is that it is an early sign of response to treatment, that the melanoma-derived blood, the derived DNA that gets out of the melanoma cells into the blood can be measured, and it will be an early sign of treatment.
Okay, thank you very much for your presentation and for coming along tonight. We really appreciate hearing, because I know you're passionate about clinical trials. So thank you very much, Grant.
No, my pleasure, and thank you to MPA for putting this on for the whole community, and everyone for their for the excellent questions that I've seen come up on Q&A. So thanks everybody. Pleasure to be here this evening with you.
Thank you. Okay, well, look, thank you everyone for those great questions. As Grant just said, it is time to close. And on behalf of MPA and our guest speakers, Professor Georgina Long, Professor Grant McArthur, and our patient speaker, Narelle Vish, a big thank you for coming along tonight. Now, one more thing, I'd like to remind you all that this is a 4-part series symposium. Our part 4, our final webinar, will be at the end of November, and we'll have guest speakers discussing the latest melanoma research, targeted therapies, and brain metastases. We will automatically send out the link to you for you to register. So good night and take care, everyone. Thank you.